Hi, my name is Aashvi, and I am a master's student in the dry bean breeding lab at the University of Guelph. And my research is focused on developing genetic markers for anthracnose resistance in small-seeded dry beans in Ontario, specifically resistant markers for the race 73, which is the most prevalent race in Ontario. So Ontario is actually the second biggest producer of dry beans in Canada, and out of all the different types of dry beans we produce, the small-seeded dry beans such as navy beans and black beans are the most exported out of Ontario. So anthracnose is a seed-borne fungal disease caused by the fungus Calotridium lindamucianum. And if you look at a plant, a bean plant, and you see dark spots on its leaves and pods, that's likely anthracnose. This fungus loves cold and wet conditions, which are pretty common in southern Ontario, and it can cause up to 100% yield loss. Not only can it cause up to 100% yield loss, it is also prevalent. If infected tissue is left behind from previous growing seasons, it can cause infections in future growing seasons. So anthracnose is currently managed by growers by just getting rid of seeds that look infected, also the use of fungicides and resistant cultivars. As I mentioned, one of the ways for growers to deal with anthracnose is through the use of resistant cultivars. These cultivars stack different genes together in the hopes of providing the plant a way to avoid anthracnose infections. But this has not been enough in high-pressure environments for the fungus. So the fungus has still been able to escape this resistance and infect plants when it is the perfect environment for it. And so there's a need to develop even more markers so they can all be stacked together in resistant cultivars. Anthracnose resistance genes in dry beans are called cogenes. Many cogenes exist for resistance against the race 73, and some of these are CO1 and CO4. Usually cogenes are part of gene clusters, and they're usually multi-allelic. So that means that for one gene, there are many alleles. And usually these alleles are part of resistant pathways. So in fungal detection, specifically anthracnose detection and defense. I plan on using a genome-wide association study to discover new markers for anthracnose resistance. So I'm using a population of 207 unique genotypes, and I'm going to be tracking phenotypic traits for these genotypes, such as height or yield and maturity, and others after they've been inoculated with anthracnose, race 73 of course. And I'm also going to be collecting genotypic data in the form of genome sequences by GWAS. So through my study and the discovery of these new resistant markers, they will be eventually bred into current elite cultivars. And as well, because this information is going to be public, other breeding programs can also use this for their cultivars. So currently I have 1 year's worth of phenotypic data from these 207 genotypes for the year 2025. I have also extracted the DNA from the 207 genotypes, and they have been sent out to be sequenced. Hopefully I will be getting those sequences back, and then for the year of 2026 I will be collecting more phenotypic data, and finally I will do the GWAS and I will find some great resistant markers for anthracnose race 73.